Thank you very much, and thank you very much to the Society for asking me to be part of this session. I'm really very honored to be here, and I'm delighted to have the opportunity to do a little reflective thinking over the last 40 years. When Brian asked me to be part of this session and said, I want you to look back over 40 years, I wasn't quite sure whether to be flattered or not. Because it's the type of thing that, somebody maybe asks you to do when you're at the end of your career, and I figure I'm only just starting. So, but at any rate, I appreciated the opportunity to do this, and thank you all very much. So I'm going to change pace a little bit, and I'm going to change scale a little bit, and we're going to try this knob and see if it works. Yes, there it is. And I do want to start off by pointing out that, is it the middle button for the pointer? Yes, there we go. Actually, I'm going to use this one. I'm going to point out that Brian actually missed a cultural phase in his pre-lunch presentation. And the cultural phase that he missed was the early Brit phase, as demonstrated here, which is typified by the absolute conviction that rain gear is the only appropriate gear for wearing at any particular social event. And you can see that I'm demonstrating that here. However, within a couple of years in Canada, I'd changed, and I'm not sure what you'd call this, early geologist phase, perhaps, something like that in 1979. At any rate, 40 years, I've been here 39 years, and I got straight off the plane, and I went straight into the mountains, and I started doing paleoecological work. So I feel that I'm pretty well qualified to talk about 40 years of paleoecology in Alberta. And as I was doing this, I've spent quite a bit of time going through all the literature and looking at all the different publications and trying to get a bit more of a handle on what has been going on in the last 40 years. And as you may know, I've done a couple of annotated bibliographies, which has also made me look at the literature a lot. And so it's a good chance to sort of sit back and see what lessons we can learn from the literature. And so here's my starting point, looking at 1975 and then looking at 2015, and looking at what paleoecology tells us over that particular time interval. And why paleoecology in an archaeology conference? Well, I like to say that paleoecology and paleoenvironmental studies put the stage behind the players. In other words, the archaeology deals with the people and what the people are doing in the landscape, and the paleoecology really tells you about what kind of landscape they're living in. It's like the dancer and the dance. You can't separate the dancer and the dance. You cannot separate the people from the landscape in which they're living. And the paleoecological work gives you that landscape and helps to provide that context, not only what the vegetation was like, but also what the climate was like and what possibilities, what opportunities the landscape offered to people who were occupying it. And as Jerry so forcefully mentioned this morning, this was not an area in which people passed through. This was an area that was home to people and has been home to people for thousands and thousands of years. And so it is important to understand the scope of the changes over the scope of human occupation in Alberta. In 1975, basically, there were two kinds of indicators that were used to do paleoecological studies in Alberta, and the two were inferences from pollen studies and inferences from fossil remains, vertebrate fossils in particular, and especially large animal fossils. And if you look at the literature and you look at the studies that were published at the time, these are basically the only two types of studies out there. There were the odd other thing that no doubt someone will point out to me, but this is my starting point. By 2015, we'd started to use a lot of other indicators. And these indicators are all what I call proxies, and I'm going to use this word a lot. A proxy is not something that necessarily reflects climate or environment in itself, in the sense of a thermometer or in the sense of a record of rainfall, for example, but it is a biotic indicator that reacts to or reflects the larger environmental conditions. So I'm going to use that word proxies. All of these are proxies for environment, for landscape, and for climate. And by 2015, we had a lot more proxies that we were using. So besides pollen and vertebrate fossils, we were also looking at charcoal, looking at sedimentary pigments, we're looking at diatoms, macrofossils, that's plant macrofossils primarily, and mollusk remains in sediments that were collected from cores and from archaeological sites. And in addition, there's a whole related field of work on tree rings, paleoecological work on tree rings, especially dendroclimatology, and work on streamflow records, for example, and on precipitation records inferred from tree rings, which is a whole separate field and really spans only the last 1,000 years of the postglacial. So I'm not going to concentrate very much on that. I'm going to concentrate on these. So the fact that the range of indicators has expanded very considerably also means that the range of expertise has expanded very considerably, and means that the range of practitioners has also expanded considerably. And what this means is that one person can't do it all, which is something I try and remind myself quite often. But that you need different people with different expertise, and that will come in when I talk a bit later as well. The other thing to remember about these various proxies is that they have different resolutions. In other words, they're telling us about different aspects of the climate and different aspects of the landscape. We already talked about things like tree rings, for example, which fall into this area here. Tree rings are particularly good as proxy records because they give us annual resolution. And a lot of the other kinds of records, like documentary records and instrumental records, also give us what we call high resolution information. Most of the records that I work with sit up here at this level here. So these would be proxy records that are based on pollen, on plant macrofossils, on diatoms, on sedimentary pigments. These records tend to span the entire postglacial, whereas these don't, but they give you less detail. In other words, they are less resolved by and large. So they give you records, or they give you information on the scale of decades, if you're lucky, or on the scale of centuries, if you're less lucky, very rarely on the scale of subdecadal resolution. And this has important implications when you're thinking about how people live in the landscape and how people use the landscape, because people think on the scale of year to year, or on the scale of a human lifetime, or on the scale of a generation. It's a very different kind of resolution. So we're going to not really talk anymore about vertebrate fossils in this talk. There is a lot more vertebrate fossils, and we have good records at the Royal Alberta Museum of many vertebrate fossils. Besides the ones that are recovered from archaeological sites, there are also paleontological studies of vertebrate fossils, and they have a wide distribution through Alberta, many from gravel pits, many from river fluvial situations, some from caves, as has already been mentioned, but they have a slightly different interpretive nuance, and they also need the background that is given from these other records in order to give them context. So I'm going to cut those out, and I'm going to cut out tree rings, and I'm basically going to concentrate from here on in on this particular suite of records. I thought I'd show you a little bit about what these look like. The ones on the left are pollen, and the ones on the right are plant macrofossils. Most of the proxy material that we work with is botanic, is plant-based, and most of it is fairly well preserved. The pollen grains here, you can see, are tiny. They are only less than 1/10 of a millimeter across in most cases. Seeds are much larger. They tend to be, this is about 2 millimeters across. This is a seed of bulrush. And you've got both ends of the plant life cycle here, if you like, the pollen that starts off the initial generation and the seeds that start off the next generation here. So they're comparatively well preserved, but they tell you different things. Pollen is adapted to dispersal across the landscape. Many pollen grains are adapted to wind dispersal, such as the ubiquitous pine grain here. And these are therefore spread across the landscape and accumulate in sites such as lakes and wetlands, and give you a regional picture of vegetation and of landscape history. On the other hand, seeds and other kinds of macro remains, molluscs for example, are more local. and they don't tend to disperse as far across the landscape, and so they give you a pattern of what's happening immediately around your site. So by looking at both of them, by looking at pollen and looking at seeds, and by looking at other proxy indicators as well, you can get a picture on a wide scale and on a very local scale, but both on a fairly coarse resolution, primarily. Here's some macrofossils. This is where I spend most of my time, and this is what I actually love doing, is looking at seeds. These are some seeds from the Wood Bog site up near Grand Prairie, which is a site that I investigated and have continued to work on. And you can see that they're quite large. This is 0.5 millimeter, so these are 2 millimeters. This is a seed of pondweed, sedge, Hippurus, mare's tail, and dock. This is actually the encasement of a seed. The seed is actually inside, slightly larger. This one's quite beautiful. They're all beautiful, and they're all really good to look at. But the fact that I have put actual species names on these, actually I haven't, it's got cut off, but anyway, the fact that you can get these down to species also tells you something else important about these kind of proxy indicators. Pollen has fairly low taxonomic resolution. In other words, you can identify pollen grains to family, or you can identify them to genus. Very rarely can you identify them to species. Seeds, you can usually identify to species, almost always to genus, and very occasionally you can only get them as far as family. In other words, the level of detail that you can get from seeds taxonomically is also much greater than you can with pollen. But I think what these two slides show you is that in order to be able to do this work, you need a great deal of identification expertise, and you need a great deal of taxonomic expertise. And with recent retirements and with people moving out of the discipline, that level of expertise has really diminished greatly in the province. And so that is one way in which over the last 40 years, I think, rather than going in a positive direction, this kind of work has gone somewhat in a negative direction. Here are some molluscs. Also from the Wood Bog site, and they're again quite beautiful. In terms of taxonomic specialists that can deal with mollusk remains, there's only one really, well, two if you count me, in Western Canada that work on these, so there's even fewer people that can actually do this work. The other point to bear in mind about these kind of records is that the kind of places where they are best preserved is not necessarily the kind of places where you're finding your archaeological sites. These records, especially continuous records, are best preserved in lakes and in wetlands. And so that is where much of the focus remains in terms of paleoecological work, is on lakes and wetlands. And here you see Daryl Beresiak and Robin and myself up in northeastern Alberta in 2010 doing some coring. But you can distinguish, I think, two threads in the records over the last 40 years. One is the study of lakes and wetlands, which I've already mentioned, and that continues to be our main focus. That also varies, though, because large lakes give you different records than small lakes, and wetlands give you completely different records to lakes, so you still have to choose your site fairly carefully. But then there is also a suite of studies that are focused on archaeological sites themselves and on remains that are recovered from archaeological sites. These may be sort of continuous samples through an archaeological site, and this is me looking really, really happy because I was out of the office and in the field, and this was so much fun. In September 2014, Heinz was kind enough to invite me along to Fort Vermilion, and we're doing some sampling on the wall of one of the excavation units. Oftentimes in archaeological sites, what you have is samples from cultural levels, or samples from feature fills, or samples that are associated with archaeological components in the site rather than continuous samples, because often the preservation locales are not as good for the kinds of paleobotanic remains that we're looking at. And that means that the archaeological site remains are often discontinuous, and they're sort of more like spot samples. The other thing that you also have to remember with archaeological sites, and one of the things that comes up a lot, is whether the plant remains, especially macrobotanical remains, are there because people are using them, or because they are simply ambient in the area around. And there are very few studies, in fact, that look at the area around the site itself to assess what the input of macrobotanic remains is to that area around the site compared to the material that is being used in the site. Now, for our particular area, we've heard mention of corn use, for example, in some of the talks this morning. And so for our area, if we found corn kernels in an archaeological site, I would immediately say that that had to be there by human agency, because corn does not grow as part of the Alberta flora. But if I find choke cherries in an archaeological context, is that because they were there, because people were actually using them at the site, or are they there simply because there's choke cherry trees growing all the way around and those choke cherry seeds got into the site adventitiously? So there's a lot of nuances, a lot of complexity to the interpretation of these archaeobotanic records. So these are some of the kinds of contexts that have been studied. Pollen and macrofossils tend to give you information about materials that are outside the site. So for lakes, things like diatoms, pigments, and ostracods are telling you about the lake itself and its chemistry and its water quality over time. And so that gives you information about lake levels, for example, and water quality, which can tell you about climate, whether it was high evapotranspiration and drought conditions, or wetter, more wetland conditions, and whether it was saline or non-saline. Archaeological sites, in features, you get things like charcoal and some plant macrofossils. Carbonized macrofossils are extremely hard to identify and can be quite challenging, and so you often get much lower resolution in terms of your identifications here. In the matrix, around the features, you can also find macrofossils. You often also find molluscs. We've done work at the Stampede site with Jerry, for instance, looking at macrobotanic remains from there. And the main remains that we found in those samples were not seeds, but were molluscs, lots of really great snail shells. You might find some charcoal from forest fires, and you often also find things like insect remains, so very different sort of suite of remains. So, looking at the literature, what do we see over the last 40 years? Well, I've put together a sort of series of paired statements here that I think summarize, and I'm going to have to turn around a little bit to read this, paired statements that summarize, I think, the changes that I see in the work that's been done over the last 40 years. Mostly in the early studies around 1975, people used a single indicator. They used pollen, or they used something else, vertebrate remains. Now we tend to use multiple indicators from a site, so you might do several kinds of proxies. Generally, people would do one single site to get a regional record. Now we tend to do multiple sites from an area to get a better handle on regional variability. The landscape is variable now. Clearly it was in the past. One site cannot capture that complexity. Although the early records did very coarse resolution sampling, often on the scale of a century or more, very coarse sampling throughout the records. Now we sometimes do very detailed sampling, even at the scale of a millimeter or more on records, which gives you a higher resolution, but still not really good resolution. In the past, we tended to do the whole record. So when you did a core, you basically sampled and did the whole thing. Now we tend to be much more selective, and we may not do the whole core. We may simply do a specific portion if that is the portion that we're particularly interested in. In the past, we did very few radiocarbon dates. Radiocarbon dating has always been the thing that we've been doing. Very few dates, and they tended to be basal dates, which were highly problematic. Now we tend to do many dates, and we tend to do many different kinds of dating methods in order to get a much better chronology and see we don't make the assumption that the deposit builds up uniformly over time. So we've gone to several chronologic methods. Here we used to do single site reconstruction, so you used to infer from the site the region. Now we tend to use multi-site reconstructions, use many sites to infer something about the region. We used to see climate as the driver, and this is something that connects very nicely with some of the talks that have gone on earlier this morning as well. We used to see climate as the driver for all changes in the paleoecological records, and if you read early paleoecological literature, you will see that used as the explanation for any change that was seen in a record. And people said that, well, there's no evidence of human activity in the paleoecological record. Well, that's because you haven't looked for it. So you need to step back and look for other explanations. And I think now we have a tendency to look for multiple explanations for the changes we see in pollen records, instead of simply saying, oh, my goodness, there's a little wiggle in the pine curve. That's got to be climate. Well, maybe not. There's more nuance. In the past, we used to have a single researcher, one person, one site. Now we tend to work in research teams with many people working on a single site. The thing that hasn't changed over time is that this is extremely labor intensive. Here's me working in the lab, sieving macrofossils, very labor intensive. It requires many hours at the microscope, and it requires really good reference collections and really good material. At the Royal Alberta Museum, we have extremely good reference collections, if any of you want to come and work with them. We have around 2,400 seed samples, for example, and almost 1,000 pollen reference samples. And you're very welcome to come and use them if you're doing a study of this type. So what do we show? So I've got like 6 minutes left, I think, so I'm going to go whip through here and give you some idea as to what this all shows. In 1993, I put together a paper for the CGA, so that's almost exactly halfway in this interval. And I looked at the number of published sites in Alberta, and you'll notice that this pretty well cuts off in Alberta. And although we're concentrating on Alberta, it's very important to remember that the biota doesn't recognize boundaries. And so although we're talking about Alberta here, many of the stories that we're talking about go well beyond Alberta. We need to look at the much broader region beyond Alberta in order to set a material into context. Many of the sites were unpublished. The data was of variable quality. There was very limited dating control, mostly basal dates. Raw data was not available, and so if you wanted to reanalyze those sites, you couldn't because the data wasn't available. And the specimens and the samples were often not curated and not in a repository, so if you wanted to go back and check the taxonomy, you couldn't. And many areas, as you'll see, were not well sampled, especially in the north and in other areas. And you can see that samples tend to follow major highways or places where access is easier. I looked at the literature to get an idea as to how this was reflected in the literature, and this is a little picture that I put together of that. These early publications in the 1940s and 1950s were by a guy called Henry Hansen, and I would actually like to sort of erase them off the whole record because they're not very good records at all. But if you look in the early 1970s, there were very few published records. The one that was published there in 1970 is the Lofty Lake record by Sigrid Litchi-Fedorovich, which has become very much a foundational record for our understanding of Alberta paleoecology, and in my mind still is a very good record. And then you can see the explosion of publications in the 2000s and in the late 2000s and in the 1980s and the 1990s. Most of these are peer-reviewed publications, and there's also a considerable amount in the grey literature, which is predominantly theses, some of which were never published or never made available, and so basically remain unknown to most researchers. And you can see that things really drop off in the early 2000s. There are a number of reasons for that. One is that some of us who should be publishing aren't, and I will turn my back at that. Jerry's laughing at me. And the other reason for that is that there's retirements. People like Charlie Schwager and Len Hills basically retired, and they haven't been replaced in the university. There is now in Alberta just one person who will teach you pollen or teach you macrofossil work, and that's me. And I get people coming from Alaska, from Calgary, from Saskatchewan to come to the lab and work with me. So there's very few opportunities for training. There's 1975. So you can see when the society was founded, this was just the start of the golden age, as it were, for paleoecological research. Sites sampled in 2015, here's the record. The blue ones are mostly on the prairies in the Scape Research Area, and the red ones are mostly in the mountains and the boreal forest. And actually the situation hasn't improved a great deal. We now have 113 sites. Some still remain unpublished, in fact many do. The data are still of variable quality. Recently studied sites generally have much better dating control than the older sites. And raw data are now available for some sites, although still very few of them have curated samples in repositories where people can look at them again. But many areas are still not well sampled, and you can see that for the whole of the boreal forest. The big difference, one of the big differences, one of the real changes, besides the transfer to sort of team approach, is also the willingness of researchers now to share data, and data being put in large data repositories. And this is a change that has been driven by funding agencies and being driven by government commitments at various levels to open data, to open access, and to share data. And there are some great repositories. This is the one that I use. It's Neotoma, which is run by funding through the National Science Foundation in the US. And you can put raw count data there to these data collectives. It's a means to archive it, so it is permanent. And that's one of the things I found when I was looking for raw data from studies, is that often the raw data aren't there. And the data can be used in other reconstructions and models, and that is the other big change as well. So people can go into Alberta, for instance, and here's an extract from the Neotoma database that shows the sites in Alberta. There's 21 of them with raw data. Many sites still are not submitted to Neotoma. Coverage is very uneven, so you've got a fair few sites in the south, but not many in the north, and it still is only a subset of data. But it means that these data are now available for people to model. And I'm going to whip through these very quickly. But these are the kind of regional models that people can now do. And this is done by Art Dyke. And I mentioned Alberta is only part of the big scene. And he put together a reconstruction looking at the changing vegetation types. This was done in 2005. So just at the end of that golden age, really, the reconstruction would not change very much for Alberta, at any rate, if we did it again. But you can start to put together these regional reconstructions, and all the dots mean these are pollen sites, these are fossil sites, and you can start to put together a regional reconstruction showing how vegetation has changed over time based on many sites, not just on one. This does a couple of things. One is it puts together this regional pattern, so you can see these broad-scale patterns, but it also shows you where the anomalies are, where the gaps are. And it shows you where things are that are different to other things. In other words, it shows you where some of that variability, that hidden variability may lie. And so this is extremely valuable work, and there are a number of people who are doing reconstructions of this kind, and I certainly commend them to your attention. So we've waded through a lot of information and a lot of data. Where are we now? Well, we're at a time now, I think, where we shouldn't be complacent about the level of data that we have. We still need a lot more data in order to be able to provide archaeologists and people doing human history with the information they need to set the people in context on the landscape through time. I think we've seen a lot of patterns that are really consistent across a broad area that help us to have some understanding of the resources that are available for people in different areas of the landscape at the past. And I think looking forward into the future, we have also the prospect of new techniques. Somebody mentioned DNA. There are studies being done now on extracting ancient DNA from lake cores, and I think those have the potential also to greatly extend the range of interpretations that we can make. So we have, we have a good deal of data. We've done a good deal of work, but there's no reason to be complacent, and I think there's an awful lot more for us to do. So I'm actually looking forward to the next 40 years. And, I hope you'll invite me back in 40 years' time, and I shall walk in here on my Zimmer frame and tell you all about what I've published in the last 40 years. At any rate, this is where we are. This is kind of how we're going on. There's a lot of things we still need to do. And one of the biggest things, I think, is this whole time scale. That the time scale of many of these records is on the still fairly coarse, and we need to scale down these records so that we can understand the changes on the scale of a human lifetime and on the scale of a human understanding of the landscape and movement across the landscape, which is going to be very difficult to do, I think, but is something that we certainly have talked about a lot, and I think we can move on into that area. Oops. And I jumped my last slide, never mind. I would like to thank you all very much for listening to me this afternoon, and it's been really fun to sit back and take a look at 40 years and filling in the gaps. Thank you very much.